Hello, good afternoon, everybody. Hello, my dear
Good
students.
afternoon, sir.
Good
Good
afternoon.
Good
afternoon, sir.
afternoon, sir.
Yeah, yeah.
Good
Good
Uh,
afternoon.
Good
yeah,
afternoon,
it is
sir.
the
Afternoon,
yeah,
sir.
yeah. Thank you. Today, it is the practical aspect, the demonstration regarding the instrument spectrophotometer. Yesterday, we had learned or heard about the colorimeter from our colleague. The next step, the instrument that will be used is spectrophotometer. When you were here, that time you had undergone attending the practicals like normal urine analysis and abnormal urine analysis, which you are finding the things which are present just qualitative, means you are just mentioning whether that component is present or not. That is the qualitative. But when the aspect comes about finding out if it is present, then how much amount of it is present, which we say as a quantitative estimation. So, in this regard, yesterday you have seen that how a colorimeter works. The same step only. This is another ahead of that basic principle procedure which we can use or make use of this instrument more elaborately to find out more precisely the values of the many investigations we do, that which is present in that any substance or blood or urine or any body fluids which we practice in our clinical biochemistry. So today we will introduce the spectrophotometer and what is the uses and its concerned subject. Okay. So it is an instrument that measures the amount of photons or the light absorbed after it passes through the sample solution. You must have got an idea what is the colorimeter and how it is used or it has been operated. In that one, we take the instrument will be having an galvanometer where you can set it to the 0 or 100% transmission, and then you are setting it with some reference solution which we often call as the blank, and then we run one sample or measure a sample which we considered as the standard, and then giving the same treatment to that standard and the test. After that one, that also we measure and see the changes or the difference between the standard and the test, and we calculate and we report it. So in that aspect, we make a light path to pass through the solution that will be measured, and these instruments they measure how much amount of the light is passed or how much amount of the light is absorbed once it is passed through it, and how much amount of the light is transmitted after absorbing there. So it is the one which there will be some relation between them. That is the amount of light absorbed, and it is directly related or proportional to the concentration of the solution. In that one, we use another part that is called as the filters. Yesterday you must have seen that one. So those filters will be either blue, red, green, like that one. So we select those things depending on the maximum amount of the light should be absorbed, or which the filter will help for that one, for the absorbing, that filter we use. So in other words, that will be called as the wavelength. That is will be called as the wavelength. It is a device to measure the intensity of the absorbed light as a function of the wavelength. So it is a device that quantifies the amount of light transmitted through the solution. So in the colorimeter there will be three parts. One will be the place where you are keeping the solution, which will be measured, and it will be attached with the required filter and galvanometer, and light will be passed through that, and depending upon the amount of light transmitted passing through that solution, that will be measured by the galvanometer. The same thing is getting applied in the spectrophotometer will be, and how much it changes, that we'll see in the next slides. To obtain the best precision and accuracy for the concentration of solution, it is equally important to select the ideal wavelength. So we all know that it will be informed to you that which filter should be used or which wavelength should be used. Somehow that should be found out how it can be done. You can see that when you look at the spectrum of the color that will be found when a white ray is passed, it will be having the range from 200 to more than 800, 900 like that. But in that one, you can divide them or classify them into the three things. One is the ultraviolet range, second one is the visible range, and then infrared, like that one. So the human eye can detect the light spectrum ranging from 400 to 700 nanometer. This is the one which we can call as the visible range. But there will be other wavelengths or the space is there which cannot be seen by the naked eye. But in this spectrophotometer, you can measure the required test for that one if the solutions are colorless, and if you want those wavelengths, that also can be measured. Major difference between colorimeter and spectrophotometer is that in the colorimeter you can measure only of the visible range, but in spectrophotometer it is not that way. It is both the things, means visible range as well as UV and infrared. Okay, here it is there. You can see that first is the gamma rays, X-rays, ultraviolet rays, and then there will be some gap between this one, the left and right, which you can call that as the visible light or visible range, which you can see that things are the colorimeter can weigh, but the spectrophotometer is the one which can measure this ultraviolet one and infrared also. So this is the major advantage of using the spectrophotometer. That is the first thing. You can have the idea that wavelengths, how it ranges from gamma rays to the short wave or AM, like this one, means higher energy to the lower energy. So we can get concerned with these three things, that is ultraviolet one, visible range, and infrared wavelength. These three things can be get applied to the at present your level. Okay, in the colorimeter, what you have done, you have seen that some filters will be kept there, known filters, and the light will be made pass through that filter, and that will fall on the solution, and the intensity of the light will be measured. So just for your information, when a white light is made to pass through a prism, which will be used in the spectrophotometer, it gives the range of the different colors. That is, you may remember in the high schools you must have learnt it, that it is called as the Walvekar, that is violet, indigo, blue, green, yellow, orange and red. So from this lower side it is UV side, and at the other side it will be, after the red it is infrared one. Okay, so based on this color of the solution and color of the filter which is used in the colorimeter, these are the set of wavelengths that are kept here. You can see that, for example, for the blue colored solution, we use the filter of the yellow filter, which will be having the wavelength 575 to 600 nanometer. For example, if it is red one, it is blue green, it is 495 to 505, like that. And if it is of the yellow color solution, it is blue and wavelength 430 to 475. In the colorimeter, what you have got, the fixed filters will be there, that it will be the filter of particular wavelength. It will be, suppose if it is of 600, it will be only the 600. But in the spectrophotometer, what can be done? The advantage, one more is there, that you can scan the wavelength. You can scan the wavelength. Suppose you have got a solution of having the violet color, you just keep it and scan as the instrument for the scanning purpose, and it will scan it and it will detect which particular wavelength it has got the maximum or the peak level of the absorbance. So that range could be between 555 to 575. This is the one more advantage with the spectrophotometer. Now, it utilizes the dispersing elements, that is prism, instead of the filters, to scan the large wavelength region. So it is a method to measure how much a chemical substance absorbs the light by measuring the intensity of the light as a beam of the light passing through the sample solution. So it is an advanced step of the colorimeter. Just advanced step of the colorimeter. In the colorimeter, you can use only the recognized fixed filters, whereas in the spectrophotometer, you can yourself only can scan which wavelength is suitable for that particular solution, colored solution. In the colorimeter, it is absolutely necessary that the measuring solution should be a colored one. It should be a colored one. But in the spectrophotometry, there is no need for that solution should be a colored one. If it is also a colorless one also, then it can be measured. And depending upon the guidelines given by the spectrophotometry, when you ask it for the scanning of the wavelength, that wavelength only you can make use and measure the things. That is the one advantage with the spectrophotometry. So let us come to some basic theories there. Each compound absorbs or emits the light over a certain range of the wavelength. The light absorption is directly related to the concentration of the compound, and as the concentration increases, light absorption also increases linearly. It means once the linearity gets affected, then the calculation goes wrong or your value will go wrong. So it should be maintained there that as long as the concentration increases, the light absorption also increases. In case if the concentration will be more and that absorption, light absorption by the filter is failed to be recorded, then you will be compelled to dilute the concentration and set it in such a fashion that it can be read with the provided wavelength. But in the spectrophotometer, usually you will have a wide option of the wavelength to be measured. The absorbance cannot be a limited one. The absorbance range can be from 0 to 2.5, whereas in the colorimeter, the absorption capacity or the range limitation will be hardly up to 0 to zero point 0.8 only, zero point 0.7, 0.8 right only. So the advantage over the colorimeter by this spectrophotometer is that it can have the wide option, the range of the absorption. So the basic thing goes that as the concentration increases, light absorption increases. As the concentration increases, the light transmission decreases. Means just imagine that you have got some colored concentrated solution and you are making a light to pass through that. When it passes, some amount of the light will be absorbed by that solution. So absorption of the light is depending upon the concentration of the solution. So they go hand in hand. As the concentration increases, the light absorption also increases. So when the light get absorbed after passing through the solution, the coming out of the test tube or the cuvette, what we call, that will be depending upon how much amount of the light is absorbed. So it means indirectly it will be opposite to each other. Means as the concentration increases, outcoming light or the transmission light, it will decrease. This is the basic thing that you should remember. Okay. Based on this one, yesterday the master told you, just I am repeating it if it is not that way. There are laws of absorption is there. The absorption of the light by any absorbing material is governed by the two laws, Boger, Lambert law and Beer's law. So they combinedly they called it as the Boger Lambert's law. This was suggested by the Boger in 1729 and it often attributed to the Lambert. That person was working more about it, and that's why it is called as the Beer-Lambert law also. This law states that the amount of the light absorbed is proportional to the thickness of the absorbing material and is independent of the intensity of the incident light. Second law is that it states that the amount of the light absorbed by a material is proportional to number of absorbing molecules. Means the concentration of this one is proportional to the light absorbed one. So that will be given by one formula like that one. If it is hello, please mute it. Now, what are the uses of this spectrophotometer? Colorimeter can be used to measure the things like which are in the only the visible one. Remember always, colorimeter is usable for only the measuring of the visible one, whereas spectrophotometer is the one which can be used for the calculation of visible light, UV light or infrared. So, making use of this one in the clinical biochemistry area or in other laboratories, it can be used for the measurement of the substrate or substance concentration like the protein, DNA, RNA, growth of the bacterial cells, and more importantly, enzymatic reactions. You must have heard the name of these enzymes like the SGO, DSGPT, or alkaline phosphatase, LDH, CPK, CKMB, all these things can be measured on the spectrophotometer, and that measurement will be called as the kinetic one. So the estimation of glucose, urea, creatinine, all these things can be measured because they at the end point they form a colored solution. They form the colored solution. Whereas the enzymatic test done for the estimation of this SGOT, SGPT, ALP, these are all of the no color or slightly the yellowish tinge color will be there. So these colorless can be measured on the spectrophotometer. So the measurement of these things are classified as the two things. One as the endpoint reaction and another one is the kinetic reactions. So in the endpoint one, what we measure? We measure the concentration of the product that is formed in the solution. Whereas in the kinetic one, we measure the rate of reaction per minute. What will be the change? It will be happening. What was the amount of that and substrate acting upon the enzymes? You must be knowing that one. So substrate and the enzyme, they combine together, make a complex, and then finally the product will be, what will happen? Product will be formed. So during that time, the change in the activity or the increase or there may be the some type, it will be decreased. That will be measured as per the reaction per minute. That is called as a kinetic reaction. So that kinetic reaction also can be measured in the spectrophotometer. This is the another advantage of the spectrophotometer comparing to the coloring meter. Okay, it can be used to measure different concentration of different molecules in the solution. So in the clinical applications, it is used to examine blood and tissues of the clinical importance, or for the sake of clinical diagnosis, you can make use of that one. Means when we are measuring these enzymes, are these some investigations like protein, albumin, inorganic phosphorus, calcium, that time they are helpful to convey the message to the clinician that this much amount of that component is present in the blood of particular person or the patient. So it helps him to make the diagnosis and accordingly to treat the patient. So for all these things, spectrophotometer is the one of the important aspect in the laboratory. There are several variations in the spectrophotometry. Of course, in some advanced laboratories, you may find atomic absorption spectrophotometry or atomic emission spectrophotometry will be there. But routinely, such things will not be made available in the laboratories. Means routinely this should not be used. Only in the research oriented laboratories you can find such instruments. Now coming to one called UV visible spectrophotometry. It uses the light over the ultraviolet between 185 to 400 nanometers. Some investigations are there like estimation of T3, T4, which can be measured at this range between 185 to 250 only. And for your knowledge, you must be heard that of SGOT, SGPT, AST, they are measured at the 340 nanometer, whereas alkaline phosphatase, it is measured at the 405 nanometer. The visible range starts from the 400 to 700 one, wherein you can measure of these like the glucose, protein, albumin, calcium, magnesium, all these things can be measured in this range, which we call as the visible range. And another one is there, that is infrared spectrophotometer. So in this one, they use of the wavelength beyond the 700 nanometer. Particularly the calcium can be coming close to that one, which can be measured at 650, 700 nanometer. Okay, and visible spectrometer is, in practice, it uses a prism. This in the earlier slide you have seen that one, how when one light is passed it converts into the different colors in that one colored wavelengths. So that in that one will be selected which is related to the particular that solution where more amount of the light can be absorbed, that will be selected by the spectrophotometer. So in the visible spectrophotometer, they use a prism to narrow down the certain range of the wavelength so that particular beam of light is passed through a solution sample. In the spectrophotometry, the absorption of the transmission of certain substance can be determined by the observed color. So remember that in the visible range or in the visible spectrophotometer, there should be the color for that any solution you will be measuring. Without a color, it cannot read it in the visible range or the visible spectrophotometer. Okay. For instance, a solution sample that absorbs the light over all visible ranges, that is, transmits none of the visible wave, appears dark. When it is not accepting anything, then it appears dark. On the other hand, if all visible wavelengths are transmitted, then and everything is passing out, then it appears as the white one. So if a solution appears or it has the absorption of the red light, it appears green, because green is the complementary color to the red. That is how they select the filters for the colorimeter. The same selecting of the filters are applicable in the auto analyzers, which will come in the next slides. So there also, in the auto analyzer, the scanning of the wavelengths will not be permitted there, or that scope is not there. There also again, like the colorimeter, you will be having the specific filters which will be referred with the word called as the wavelengths only, and they will be the fixed wavelengths. So visible spectrophotometer, they narrow down the light that is passed through that one, and it will be seen that one the maximum amount of the light is absorbed in the visible spectrophotometer. Okay. You can see that how this one happens. At the left side, the light source is there. It can be in the colorimeter, it will be just a tungsten bulb, whereas in the spectrophotometer, it will be the halogen lamp also. When the light passes through that one, the lens will be there. From the lens, it passes through to the prism. So this prism, when it passes there, the different colors of the lights will be emerging. But what happens? The maximum light which can be absorbed, it can be made to pass through a slit. It will be a small hole like portion will be there. From that one, it passes to the cuvette. We can call that as the cuvette or the glass test tube, wherein the measurement of the solution will be taken place, or you are keeping the solution in that one. So here the amount of the light that is passing through that, it is depending or it is related to the amount of light absorbed here. It means it is directly related to the concentration of the solution, and it is inversely proportional to the amount of light transmitted, that is coming out of this cuvette. It will be inversely proportional. Here it will be directly proportional, and this one are inversely proportional. So when once the transmitted light coming out of this cuvette, it will be digitally absorbed or detected by the digital display, and that will be measured on this display meter. You can see that in the colorimeter, you must have seen yesterday, that may be having a galvanometer. Nowadays there is improvisation is there. They are also providing the colorimeter with the digital display only. So it is the way how the spectrophotometer works. From the light through the lens, they fall on the prism. In this one, the maximum required are selected scanned wavelength that will be passed through the slit. It will fall on the cuvette containing the solution of some concentration, and it will be transmitted and it will be detected by the photocell, and it will be converted and expressed in the digital display of the meter. This is how the spectrophotometer works. Same one only here. The same, just I got another photo, that's why I am displaying. Same thing it is. So the spectrophotometer, it is same related only. The whatever I said showing the picture, it is written there. And here, when the light path will be passing, it will be measured in the centimeters. That will be measured, and standard spectrophotometer uses a cuvette that is 1 centimeter in the width. The width of this one should be a constant one, so that it will be very helpful for the measurement of the how much amount of the light is passed. And for the colorimetry, substrate must participate and must produce a color compound. As I said earlier, in the colorimeter, it should be of the visible range or the visible color which should be there, so that only you can measure the things in the colorimeter. Now, this you can have the idea of this spectrophotometer. In this spectrophotometer, this is the device wherein you can open this block, and there will be two positions will be there. Means keeping the cuvettes in that place. One cuvette will be used as the blank. The blank is the one which is consisting of the reagent that is used for the particular investigation. For example, if you are measuring creatinine, it will be having blank. The blank is the one which consists of all the materials, all the reagents, except the component of the test or the sample that should be tested one. It will be minus the sample that should be tested. That will be called as the blank. And then next, when you keep the blank, selecting the required wavelength, you should press the button here, it will be there required. So you will be asking that instrument to measure the blank. So what happens? That instrument set it 0 to the blank. Set the instrument to the 0 with the blank. Then what you will do? You will process a standard sample. Means same amount of the reagent which you will be using for the test, that will be added with some standard. Standard means a sample of known value. Suppose if it is a glucose estimation you are doing, that time you are running a standard. So that standard will be, we are taking into the account 100 milligram standard. That will be added to that process there, and we are trying to find out what is the absorbance of that one. So once you keep the standard cuvette in that one, press the button and value will be noted. And then followed by, you run a unknown sample, that as the test of the sample test. So that will be measured, means in that cuvette only. Blank one cuvette and another one will be the standard and followed by the test. This is why you can do it here. The advantage here is that in some spectrophotometer, what they do, even in the semi-auto analyzer or fully auto analyzer, taking into the account all these absorbances, it will measure, calculate and display the direct value here. That can be possible, and it is quite common feature in the semi-auto and fully auto analyzers. So there are some basic optical instrument components are there. These are, just go through them. Source, that is the light which radiant energy and desired wavelength. Wavelength selector, a device that isolates a restricted region of the electromagnetic spectrum used for the measurement. This is one. Sample container, that is the cuvette where you keep that one, that is called as the sample container. And detector, that which detects, means it find out how much of the absorbance is done, and that will be converted one, that will be done by this detector. And signal processor and readout, amplifies and attenuates the transmitted signal and sends it to the readout device as a meter, means that is the one which converts the signals that has come in the recorder of the computer one, means the absorbed light, it will be converted into the detector device and it will be expressed in a digital way. These are the some parts of the colorimeter, or which are the words that will be used in the spectrophotometer. So you can have a look at this one. The processing is that first you have to do, turn on the machine and let it sit for the 15 minutes before, means it is for the spectrophotometer, one requirement is there, you have to put it on and allow that to have the warm up. We call that as the warm up position. So first you have to start and keep it for 15 minutes in a on position only. Very first basic thing. And then place a blank in the spectrophotometer. For everything they will be providing the buttons and guidelines will be given. And once the blank is kept, you have to set the blank to the 0 by adjusting the knobs. This is a cuvette it is. So you are keeping in that slit only and having the blank, means it can be sometimes distilled water also. Just you keep the distilled water, take the reading of the reagent blank. Even suppose if the reagent itself is a colored one, for example in the case of creatinine estimation, you are using the picric acid one. Picric acid is the one which has got basically the yellow color. That time what happens, we have to subtract, means we have to see that the color of the blank is not interfering with the reagent of the test one. That's why we subtract or remove out the amount, whatever the absorbance may be there for that reagent. That's why what we do, we set the instrument with the distilled water, running that as the blank and setting that as the 0, and we take the reading of the reagent blank also. So that will be subtracted in the test or the calculation purpose. Okay. So followed by next, you will be keeping the standard and running that one, knowing that one, and followed by the standard, you are keeping the sample. So once you keep it, wait for the 10 seconds, that will be measuring the transmitted light or the absorbance will be recorded. So the procedure is that turn the spectrophotometer and let it sit for 15 minutes, blanking, note the proper volume of the sample in the cuvette, choose and set the wavelength. Suppose you are estimating the creatinine, it will be requiring of 530 nanometer, or in a language of filters if you say, that will be the green filter, like that. So you have to select the cuvette, or you can ask the instrument when we are using the spectrophotometer to scan for the wavelength, then it will suggest which wavelength should be used. Usually for the creatinine it will be the green filter. So choose it and set the wavelength of the light. Color of the light chosen should be one known to be absorbed by one chemicals thought to be the single wavelength of the light. In some investigations, they use two wavelengths that will be called as the bichromatic wavelengths we call. Usually that is very much useful in the estimation of bilirubin, where we use the bichromatic one, because it is basically a very highly colored pigment it will be there. So that color also may be interfering. What happens by using the bichromatic wavelength, you can subtract or compensate the interfering factors. In those conditions they use the the bichromatic wavelength. But usually it will be better to use the single wavelength, that is monochromatic one. So here, take the standard solution in another cuvette and insert it in the test conducting space and press the calibration. Calibration means standardization. So running the blank, note the value and then run the standard of the known value and then note the absorbance. So noting of this value of the standard, it is called as the standardization, or you can simply, it can be called as the calibration, we can call that. So once the instrument is calibrated, then with the same wavelength, one can process the test for the unknown one. Now the measurement of the solutions which you need, that can be measured. So what we'll do, how this will be exactly done, I will show with one example. So we will take into the example, just of course separately it will be shown to you, or if you come to the college, that time practically it will be made you to process and you will be more acquainted with that one. But now just I am showing the procedure and the way how it will be calculated, taking into the account spectrophotometer. It can be done with the colorimeter also, because it is the one which can be measured in the visible one. So whichever are there, the visible one, that we take into the for the estimation with the either colorimeter or the spectrophotometer. Now coming to the example of processing blood plasma, actually it is in the practice always telling blood glucose. Very earlier days they used to process with the directly the blood, whole blood. That's why that popular word has remained still using that one, but nowadays we use the plasma. So plasma glucose will be done using the spectrophotometer for this purpose. Now for that, what you have to do, you have to take the 3 test tubes, keep them, label them as the blank, standard and test, and then you are adding in all of them 2 ml reagent. That is, the method we use is the God method, we call is that one, that is glucose oxidase peroxidase method. That reagent will be there. That reagent you are adding 2 ml in each of them and taking one standard which is of the known value. That is, here we take 100 milligram per deciliter, that one, and we are adding only the 20 microliter, only 20 microliter in the only the standard test tube, only in the standard one, and the sample we are adding 20 micro, that which we should measure, that is 20 microliter. To compensate that one, we are adding 20 microliter of the distilled water in the blank test tube, that is distilled water. So mix all of them and incubate it for the 15 minutes at 37 degree, and then we will measure the absorbance of reagent blank, standard, test against the distilled water at the 530. In some places that can be measured even with the 505 also. 530 nanometer is the spectrophotometer. Now next, what will be the step? So we have, after the 20 minutes of incubation, we have taken the absorbance. We have taken the absorbance of these 3, reagent blank, standard and the test, against distilled water. Against the distilled water, keeping that as the blank. Distilled water as the blank, we are taking the value, reading it. The reagent blank is showing 0.025, standard 0.750 and test 0.459. So we have recorded these 3 values on the spectrophotometer. Now there is one formula for the calculation that goes like this way. This one, absorbance of the test, the blank absorbance is deducted in the, subtracted in the absorbance of the test. Same absorbance of the blank is subtracted in the absorbance of the standard, that is TB minus AB, multiplied by concentration of the standard, that is that should be concentration of the standard. You have seen in the last slide, it was just we have added 20 microliter. We have added 20 microliter. The concentration of this one is 100 milligram per deciliter, but we have used only the 20 microliter. Now you can see here, I have shown here, standard concentration is 100 milligram per 100 ml. We have used only 20 microliter. So what is the concentration of the standard in that 20 microliter? That it should be calculated. So I have shown here 100 ml is equal to 100 milligram. Then it comes as same concentration is 1 ml is equal to 1 milligram. 1 ml is equal to 1 milligram. It means when you convert it into the microgram and microliter, it is 1000 microliter is equivalent to 1000 microgram. This is how what the concentration is there in our test. The same thing is maintained. Volume of the standard used is only the 20 microliter. So what happens? 20 by 20, that nullifies there. So 20 microliter. So these absorbance divided by 100 multiplied by the 100. You can see the next slide. You can see here. So what I did? This is the test absorbance minus the blank absorbance, standard absorbance minus the reagent blank absorbance multiplied by the 20 microliter. 20 microgram is the concentration of the standard in the test we have used. The concentration of the standard is only 20 microgram, and effective volume of the sample, that is how much you added? 20 microliter. So the calculation goes this way, 20 by 20. So they nullify each other. So 0.434 divided by 0.75 after subtracting the blank value multiplied by 100 only. So your final value will be 59.9 milligram of the 59 milligram per deciliter is the value of the blood glucose. So this is how the standardization, calibration procedure, all this will be done on the spectrophotometer. So this estimation can be done also on the calorimeter, because it is of the one which is producing a color solution. So this color solution can be used in the calorimeter also, but more precisely in the spectrophotometer. What is the difference between calorimeter and spectrophotometer? It is, first thing is that calorimeter limited for visible portion, ultraviolet and infrared are possible in the spectrophotometer, work out very cheaply, hardly it will be, means the cost of that one is very low. It can be affordable by any person, whereas spectrophotometer is very costlier one. Here the 2 digits are available after the decimal point. It is there, whereas in this one 4 digit reading after the decimal point. It means very precise value will be available, and particularly it will be of the help in the research projects. And it is less sensitive because the any outer circumstance areas, the atmosphere or the heat outside, that can affect the light absorbance or the heat of the light that should be there. All these things will be affected, whereas that cannot be possible because it will be a closed compact chamber will be there, wherein you will be measuring the cuvette or any other thing you will be measuring. And here only the glass filters are used in the colorimeter, and here the prisms are used, where it will be more precise, where the light will be more focused and more concentrated, and the required wavelength can be selected. So prisms are of good use only. And here the tungsten lamps are used in the colorimeter, and halogen lamps are used in the spectrophotometer. And in the colorimeter, the solution that is red, a solution that should be red, the reading solution, there should be a colored one. Remember always, it should be in the visible range. Whereas the compound to be read in the spectrophotometer, not necessary that it should be of the colored one. Okay. Now, based on these things only, the auto analyzers work. Just I am giving introduction to that one. Of course, it is in this class. There is no scope for that one. But just for your knowledge purpose, I am telling you. Sample procedure gets applicable in the semi-auto analyzer, fully auto analyzer, but with some modifications. What are the changes we can find? There is no need of changing the filter. Suppose you are done glucose estimation and next you want to do the bilirubin or calcium, then you have to change the filter. There is no need there. Already the filters will be there, fixed one. Only you have to select it, making the program there. No need of scanning for the wavelengths. The same thing, everything will be the set already one, and manual application or handling is minimum. Means addition of the reagent, pipetting out the sample or the reagent, it is not there. So because by doing that one, there can be the involvement of manual errors in the estimation. So all these things will be avoided. So no blanking, standardization every time. Suppose if one batch you have done glucose, so in the morning, in the evening again you are doing, that time again you have to run blanking, standardization. But in the auto analyzer, that should be done with the colorimeter or spectrophotometer. But in the fully auto analyzers, the values of this blanking, blank or the standard will be noted there, and that can be made use of the next time when you are using. So the auto analyzer make use of that one. There is no need for you to run the blank and the standardization, so that by that you are not wasting your time or the reagent. Everything is with the programming. And another thing is that addition of the sample standard is also not required. All these things will be taken care by the instruments will be there, because the instruments will be provided with the probes which they do what the work you can do, and they can do better than you people. So another thing is calculation is not required. Applying the formula, doing that subtraction, multiplication, everything, nothing. The instrument will be ready with all the programming and taking into the account what the values have come or the absorbed. It will do the calculation and it will finally come out with the reading. So with the initial programming, all such activities can be avoided. Only thing is that once you should, in the initial stage, you should program it. What will be the type of test? What is the wavelength you are using? What is the amount of sample to be picked up? It may be 20 microliter, 100 microliter, and whatever the reagent volume that should be picked. All those things you should give the information to the instrument that will be programmed, and according to that program, that instrument will do the testing, and finally it will give only the values only. By this what happens? The time will be saved and manual errors can be controlled. This is how the measurement can be done in the biochemistry, clinical biochemistry. Initially it is colorimeter, then it is spectrophotometer, and in advanced stage it is the fully auto analyzers. So this is the picture of semi-auto analyzer. It is a semi-auto analyzer. In this semi-auto analyzer, all programming can be done, but only thing is that it can be made to suck whatever the reagent samples are there, that will be added by you manually, kept at the incubation if it is required, and then you are just pressing this button, dipping this tube, sucking tube we call this one, this sucking tube in that solution. So instrument will suck it and it will pass through their pipe and it will go into a fixed cuvette there only. So that measure there and further calculation and giving the values will be done or it will be displayed here, or it may come out from here as the printout. That's why you are mixing the sample and reagent. That part is the manual one. That's why it is called as the semi-auto analyzers. Now same thing, it is called as the fully auto analyzer. In this one you are seeing that there are two probes are there. You see this probe and this one is also the probe here. What happens? These are the test tubes which are consisting of the samples, one or two there. There you are keeping the sample, patient sample, and here the cuvettes will be there. These are full with the cuvettes. So this one will come for first in the process. Only thing I will tell you in the starting one. Here you are all, it is a computer based one. In this one you are programming. At the fifth position there is one sample of X and he needs urea, creatine, protein should be done. That indication you are giving to the instrument. Accordingly it will pick up the samples for the glucose, creatinine, protein, and it will dispense here following cuvettes. So by the time what happens, this probe will move automatically and it will take the reagents that are kept at this portion inside. These separate compartments will be there. In every compartment it will be, everything is programmed automatically. The reagents will be kept here and it opens and it dips there and take up whatever the amount of reagent, which reagent should be taken, and it knows very well at which cuvette which sample is there. It dispenses the reagent in that one. So mixing also will be done there, and it will be here only the reading filters will be kept there. It will all, everything will be fixed here. This one. So in this rotor, we call that as the rotor. Both the things are rotor. They will be under the movement only, continuous movement. So while movementing only, that will be red. That will be red. Once it read, that absorbance, it goes to the computer section. That will be recorded, and with the values of blank and standard concentration, whatever it is, way of feed it or program, it will do the calculation and it display the report directly. So your trouble or your headache is there only to mount the reagent here, load the sample and programming. That's why these fully auto analyzer persons who manufacture, they call it as the walk-away system. Just you program and go away. So by the time under 10 minutes after taking the tea, you come back and take the report. This is how they claim it. So it needs the maintenance. That one part is there. And it is very costlier also. Remember, they may be costlier. The rates may be between 50 lakhs to 80 lakhs like that one. Some semi-auto analyzers, they are costing nowadays 10-15 lakhs like that one. So this is about the measurement of the tests that are done in the clinical biochemistry. Basically it is colorimeter, and then the spectrophotometer, and then it is semi-auto analyzers, and then finally it is fully auto analyzers. So by applying these fully auto analyzers, you are spending very less amount of the time, less amount of the reagent. You can see that one in the spectrophotometer, one method I shown you. It was made that you must use 2 ml of the reagent for each test. It may be 2 ml for the blanking, 2 ml for the standard, and 2 ml for the test. Whereas in this fully auto analyzer, hardly with the 400 microliter or 500 microliter that can be processed. So you will be saving the reagent. So you are economically, you are using the reagents, so you can save it. But the cost of this instrument will be very high, and also the maintenance also is very sometimes very high. That will be the part of this one. But another thing is that with the fully auto analyzers, you can give the report in a very less amount of the time. The patients are serious or patient in the ICCU, that time they will be in the very hurry position and they will be in need of your report or your support for that one. So with the help of these fully auto analyzers, you can support them. Whereas the spectrophotometer, it has got the applicability in the research laboratories. There is no any speediness or hurriedness. No one will be sitting on your neck to demand for the report. There you can use it. But spectrophotometer, it has got its own value. Means the comparing to the colorimeter, the precision is very high. It's very sensitive instrument, and all those things are countered here, and means you can find those things in the fully auto analyzers. So this is how we use the different type of the instrument to measure the components like urea, creatine, protein, calcium, or some tumor markers, or thyroid functions, or lipid profile, whatever all those things, it can be measured here. So this is related to the quantitative estimation we do. This is one, it is not for the estimation by the qualitative method. It is only for the quantitative estimation. Okay? And then now there is some type of questions are there. Just note down this one and try to find the answer for that one. So question number 1 is, how can a concentrated sample be analyzed when it doesn't obey Lambert's and Beer law? So you have to refer to the Lambert-Beer law and then you have to find the answer for this one. Second question is that for a photometric estimation, is it necessary to always have a colored solution? That is for the spectrophotometer. And what do you understand by the standard solution? I have told you what is that one, you have to define it. And what is the importance of the blank? Why that blank is run? And now, and the last one, what are the advantages of spectrophotometer over the colorimeter? That you have to find out. In the whole this class, I kept on telling the advantages of the spectrophotometer over the colorimeter. So you prepare the answer for this one. Try to find it out. Okay. Thank you. Thanks all of you.
Thank you.
Thank you, sir.
Thank you.
Thank
Thank
you.
you, sir.
Thank you, sir.
Thank you.